Hello students, today I am with you RJ ma'am. Today we are going to start the lecture number 4. In lecture number 3 we read about the cell organelles, the heroes of the cell, and that we read about the plant main cell organelle, that is plastid, which contains chloroplast. Okay, now let us do a quick revision of lecture number 3 so that we can further move on towards our lecture number 4. So till now students, what we have studied about what is cell membrane, what is nucleus, what is cytoplasm, and all the parts of it. Now in lecture number 3 we have studied about what students, plastid. What is plastid? It is the main component through which a plant prepares its own food and stores its foods and provide colors to the plant parts. Okay, so first plastid type we studied about is leucoplast, then chromoplast. It is just a small quick revision for you all so that you can remember it well. Okay, and last one is chloroplast. Now leucoplast is the one which does not provide any color to the plant, but it is for the storage of the food, that is it in the form of starch. Next one is chromoplast. It is for providing colors to the plant, as I told you about the rhodoplast. Okay, red color, brown color, everything, the color which is provided to the plant is responsible for this thing. Now last one is the chloroplast. Chloroplast means the chlorophyll pigment, the green pigment which is used for the photosynthesis process. Till it we have read in lecture number 3. And about light reactions and dark reactions we have seen. In further upcoming lectures we will see the video of light and dark reaction, and we will understand that how light and dark reaction occurs. Okay, now let's start with our lecture number 4 about the new 2 cell organelles. Okay, so moving on, main content of today, we are going to discuss about the 2 new cell organelles of our cell. First is the double membranous cell organelle, that is mitochondria. The mitochondria is also known as the powerhouse of the cell, or it is also known as the storage battery of the cell. Now second cell organelle is endoplasmic reticulum. Endoplasmic reticulum is bifurcated into 2 types. First is smooth endoplasmic reticulum, that is also abbreviated as SER. I am writing it over here. This is also abbreviated as SER in short. And for if you write for rough endoplasmic reticulum, the abbreviation will be used as RER. And also remember this thing, everywhere I speak about ER, it means endoplasmic reticulum. It is in short form, or we can say it is in abbreviated form of this. Okay, now why there is a difference between the both types of ER, we are going to discuss this thing in this lecture only. So moving on towards our first cell organelle, that is mitochondria. What is mitochondria students? Mitochondria is known as the powerhouse of the cell. What is mitochondria? It is known as powerhouse of the cell, or it is also known as storage battery of the cell. Now what is the significance behind these lines? Why it is known as powerhouse or why it is known as storage battery, that we are going to discuss in this lecture, in this topic only. So first we should see that who discovered it and who named it. So first it was seen by Colliker, and he saw it in the flight muscles, where he saw it in the flight muscles of insects. And the term mitochondria was given by Benda. The scientist was Benda who gave the term mitochondria, and it was observed by Colliker in the insect cells. Okay, so you just have to remember the term Benda over here, because it is more important rather than Colliker, because moreover the Benda is moreover asked in examinations. Okay, now the second point about mitochondria is that it is a rod shaped structure and it is found in the cytoplasm of all living cell organelles. Now, what is rod-shaped structure? Rod-shaped structure over here represents the oval shaped in which the mitochondria is present. Okay, the shape of the mitochondria is like this. Okay, or you can flatten it more. Okay, it is an elliptical shape. You can say it as elliptical shape, you can say it as oval shape, you can say it as rod shaped also, you can say it. Okay, now this mitochondria is not present in mammalian RBCs. Mammalian RBCs, I want to tell you the fact of this thing, that mammalian RBCs, when they get matured, they lose all the cell organelles present in them. Maybe it be mitochondria, maybe it be endoplasmic reticulum. Okay, so when mature RBCs has become, they lose the cell organelles. So mitochondria as well as in endoplasmic reticulum we will discuss, it is also absent in that. So you have to remember this thing, that mammalian RBCs does not contain mitochondria in them. Okay, so you should mark it important over here, that in except mammalian RBCs, it is present everywhere in every cell. Okay, now the next point about mitochondria is they are also absent in prokaryotes. As I have told about you, that prokaryotes were the primitive cell organelles, means primitive cells they have, and primitive cell means they were less developed, so cell organelles were also not developed at that time. So it is also absent in prokaryotic cells. Okay, now the next point about mitochondria is that it is known as powerhouse of the cell, or the storage battery of the cell. So why it is known as powerhouse of the cell? Because all the energy that is synthesized inside the mitochondria in the form of adenosine triphosphate, ATP. Okay, so it is known as the storage battery of the cell, or we can say that it is known as the powerhouse of the cell. So it is a great important thing you have to remember, because in all examinations like competitive, NTSE or NSO, if you are going to fight, then this question may be asked in your examination. So it is a important thing, powerhouse of the cell or the storage battery of the cell. Okay, now I am moving towards my next slide. Structure of mitochondria. Now, what do you think so the structure of mitochondria contains? I said it is oval in shape. Now, what this oval shape contains inside it? What are the parts of mitochondria? We are going to discuss over this slide. Okay. So, it is a double membranous bound structure. First thing you have to remember this, that it is double membrane structure, means it is separated by the outer membrane and the inner membrane, and there is a space between the outer and the inner membrane. Okay, there is a space which is known as perimitochondrial space, that we are going to discuss in the upcoming diagram of mitochondria. Okay, now the inner membrane is folded. Folded means there is enfoldings of the inner membrane which are known as cristae. What it is known as? Cristae. Okay, there are enfoldings which are known as cristae. Now, the cristae are the enfoldings of the inner mitochondrial space. You know that thing which I told you, which is responsible for the respiratory cycles like Krebs cycle. Krebs cycle is the cellular respiration which occurs in the living cell organism, but we are going to discuss about the Krebs cycle in the upcoming lectures when we will discuss about the human physiology. Okay, now you remember this thing only, for cellular respiration Krebs cycle is responsible. The next point that arises is the main thing which is going to be synthesized inside the mitochondria is the ATP, and ATP stands for, students, you remember this thing, adenosine triphosphate, and they are synthesized in which part of mitochondria? They are synthesized in oxysomes. Okay, an oxysome is a part of mitochondria where the ATPs are synthesized, and it is also known as F0, F1 particle. We are going to dissect the mitochondria in a broad view. Okay, now the space between the outer and the inner mitochondrial membrane is known as perimitochondrial space, And the last part is the fluid which is present inside the mitochondria is known as matrix. As in plant cell, we have discussed about the chloroplast. I told you there, every time a matrix is responsible, a clear liquid which is present inside every cell organelle is known as matrix. In chloroplast, it was known as what? Stroma, but here it is known as matrix. Okay. Now, we are going to see the structure of our mitochondria. We are going to see the structure of mitochondria, that how does it look. Okay, so let's revise us first what are the parts of mitochondria, then we will draw it step by step. So first part is the outer membrane, second part is the inner membrane. The space between the inner and the outer membrane is known as what? Perimitochondrial space. Okay, now you remember the inner membrane has infoldings, and infoldings are known as cristae. Now cristae contains the oxysomes. Students, cristae contains the oxysomes, means these oxysomes are embedded on the cristae. Okay, now mitochondria has its own DNA also. Its own DNA which is present in the form of circular form. It is not double helical. Here it is present as in circular form. Mitochondria has its own DNA plus it has its own proteins and ribosomes. Okay, so it is known as semi-autonomous cell organelle that I am going to discuss in this upcoming slide. Okay, now let's draw the structure of mitochondria, and you should draw it with me so you can remember the structure of mitochondria. So let's draw first the oval shape. Remember that was oval shape. So first we are going to draw an oval shape, that is the outer membrane. Okay. Now this is the outer membrane. Okay, and now I'm going to draw the infoldings of the inner membrane with itself. Okay, so this is the inner membrane, students, and this has a blunt ends like this. This inner membrane is infolded like this, and it has a blunt ends. Okay, can you see it clearly over here? Okay, now we are going to differentiate between both of the two. This one is the outer membrane, and outer membrane has specific properties of it, and inner membrane also has specific properties of it. Okay, inner membrane. Now, if we talk about outer membrane, it has less proteins. It has less proteins, more lipids, and it is permeable. Permeable means it can allow each and every molecule or ion to enter inside it, okay, or to move outside it. So it is permeable in nature. But if we talk about the inner membrane, it has more proteins, less lipids, and it is selectively or semi-permeable. You are better familiar with these terms now, so I don't have to repeat it again and again. So it is selectively permeable. Okay. Now, if we talk about the space between the both of the two, the inner membrane and the outer membrane, this is the space between both of the two. It is known as what? Perimitochondrial space. Perimitochondrial space. Okay. Now, there is a particular matrix present over here, means it is a liquid and it is pale yellow in color. Students, remember this thing, matrix is usually pale yellow in color. Okay, so this is known as what? Matrix. Now in matrix there is DNA present, and DNA is present in the form of circular, and ribosomes are also present. So we can draw DNA over here. It is present in circular form. This is circular DNA. Remember students, the labeling means a lot in exams, so please label the diagrams clearly. Okay, mention the things clearly. There are ribosomes present, which I am marking with the yellow color, students, here. So it is also known as semi-autonomous cell organelle. Semi-autonomous means it can replicate itself. Replicate means what we are going to discuss in the upcoming lectures also. Remember you this thing that plastid also had its own ribosome and a DNA. So it was also semi-autonomous. So it is ribosome. Okay, now the main important part, I am going to draw it over here. Just watch it carefully. Where are the oxysomes present? Oxysomes are embedded on the cristae. These enfoldings of the inner membrane are known as what? Cristae. These enfoldings of the inner membrane are known as cristae. Okay, and they have, I am marking it with a red color over here. They have oxysomes embedded over them. It is present on each and every cristae, and these oxysomes have a particular definite structure which I am going to show you in the upcoming next diagram, and I am going to tell you how the ATP is synthesized and where and which part the ATP is being synthesized. So it is present over it. So these are known as what? Oxysomes. OXYSOMES. And this is the main important part for ATP synthesis. Okay, now did you understand the diagram of mitochondria over here? I am just going to make a quick revision of the whole diagram. First, I have drawn the outer membrane that has less proteins, more lipids, and is permeable. Next, I have drawn the inner membrane which has more proteins, less lipids, and selectively permeable. Now, the enfoldings of the inner membrane are known as what? Cristae. Cristae has oxysomes embedded over it, and they perform the function of ATP synthesis. Now there is a matrix present inside the cell organelle, and this matrix contains two important things. First is the circular DNA, and second one is the ribosome, and both these are responsible for replication processes. So it is also known as semi-autonomous cell organelle. Okay, did you understand the diagram over here? Now this diagram I am going to show you through a small video. So please pay attention here. Okay. So this is a very small video of mitochondria, where mitochondria is present inside the cell, where is nucleus present, where are the cell organelles, where are the cristae, where are the oxysomes. Okay. So I am going to just play it for 2 to 3 seconds, so you will understand, and we should learn it over here itself. So students, you can see the structure over here. This mitochondria is known as powerhouse of the cell, or the storage battery of the cell. Now you can see over here that there is mitochondria present inside the cell, and there are lakhs of mitochondria present inside each and every cell for ATP synthesis. Now you can see over here, this is the cristae over here, and these are the small ribosomes present over here in the mitochondria. Okay, now this is the oxysomal part which is present over here. This is the oxysome. You can see it clearly. Okay, now these are the enfoldings. You can see this is rotating. These are the enfoldings where the oxysomes are embedded and the ATP synthesis will takes place. Okay, now you can see here this ATP is being synthesized. These are the yellow colored ATPs which are moving inside the mitochondria after the synthesis through oxysomal part of the mitochondria. Okay, now this is the matrix. Over here the circular DNA and ribosomes are present. Okay, now this was the dissection of the mitochondria, and you can see here that how does it play a great role in ATP synthesis. And why ATP is important over here? Because ATP is the main thing which is responsible for our energy generation. Okay, now moving towards the next slide, we can see the diagram of mitochondria over here. Okay, we have drawn it also. So first is the outer membrane. Remember students, every time when you see a diagram, just see it stepwise. Do not get into the bulk, then you will get confused. Okay, first see the outer membrane. If it is not present, then see the inner membrane. If it is also not present, then see the matrix part. Okay, move stepwise. First outer membrane, then inner membrane, then intermembrane space or perimitochondrial space in the case of mitochondria, then cytoplasm or the matrix of the cell organelle, then ribosomes, then DNA, and if it is not present in that, then other cell things that is present inside the cell organelle. Okay, do not get jumped into the cell organelle and get confused over here. So first we will see the outer membrane, then after we will move towards the inner membrane. Then there is a space between both of the two which is known as what? Intermembrane space. This is labeled as here. Okay, now if we talk about the matrix part, then it is known as matrix. Okay, then matrix granules. Matrix granules means the proteins, lipids and any other thing which is present inside the matrix, they are known as granules. Okay, now the ribosome is also present. Ribosome is also present, and mitochondria DNA. DNA means the deoxyribose nucleic acid. Now crista, these are the enfoldings which are present. These enfoldings are known as what? Crista or cristae, you can announce like that also. Okay, now last is the oxysomal part which is responsible for the ATP synthesis, and ATP synthase enzyme is present for the ATP synthesis. Now where the ATP is being synthesized and how it is synthesized, that we are going to see by dissecting the oxysomal part of the mitochondria. So in the next diagram I am going to show you the oxysome part. So see it clearly students. In this I am going to draw over here the oxysome part. Now remember this thing, the oxysome part has been attached to the inner mitochondrial membrane. Okay, the inner membrane of the mitochondria. So first we will draw the part which is outer, and then we will draw the part which is inner. Okay, so look here, this is the outer membrane. Okay, and this is the membrane part on which the oxysome is being present. This is the inner membrane. You can see it like that. Okay, by turning the mitochondria diagram into a landscape form, you can see it clearly. Now this is the part, cristae part. Okay, you can see here the inner foldings of the inner membrane. So this is the cristae part. We can write it over here as cristae part, or we can put a slash over here and write inner membrane part. Okay, now what happens? This is the outer part. You can write it as outer membrane. Now what happens? This inner membrane part is divided into two phases. First is the inner phase which is inside the matrix part. This will be the matrix part, student. Did you understand this thing now? When the inner membrane is present, here the matrix part is also present over here. You can label it as matrix part so you will not get confused. This is matrix. Okay, so it is divided into two phases. First is the M phase, which is present towards the matrix part, and the C phase, which is present towards the outer membrane part. So I am labeling over it here. This phase is known as M phase, matrix phase. Okay, and this phase, which is present towards the outer membrane, this phase is known as what, students? C phase. And we can use the full form of over here. This is also known as cytosol phase. Okay, and inner membrane has some kind of proteins also inside it. Okay, so we can draw the proteins over here with a yellow color. I am drawing it over here. You can see it. These are the proteins which are present inside the inner membrane, which is responsible for the electron transport chain. So I am writing over here ETC, and this ETC is a small abbreviation which is used for the electron transport chain. Okay, now I am going to draw the main part which is responsible for ATP synthesis, that is oxysome. So where oxysome is present and how it is present, so I am drawing it with the white color only. So here is the base part. There is a protein present over it, and there is another protein present over it. Okay, now I am putting this part over here, so you can see it, this landscape part in a portrait form over here. So this part is represented as like this. Then over it a protein layer, and again there is a protein. Okay, now it has three names, means this part is divided into three names. First is the base part. This is the base part, student. Okay, this part is known as stalk part. It is divided into three. First is the base, second one is the stalk, and last one is the head, where the ATPase, means ATP synthesizing enzyme, ATPase is present over here. Now you can see this part in the this form. Now each and every crystal has many oxysomes present over it. Okay, this type of arrangement you can see when you dissect the mitochondria and you dissect the oxysome. You can see it clearly. Okay, this part is base, this part is stalk, and this part is head. Now the ATP synthesizing enzyme ATPase is present over the head. Now where the ATP is going to be synthesized and how, what will happen? So in when we read about the cellular respiration, or when we read about the photolysis of water in the plant physiology, I told you that when water splits, it splits in the form of electron, proton, and an nascent oxygen. So the protons which are transformed in the inner matrix also and inner cell organelles, or they are transferred to other cell organelles also, the extra protons. Okay, now the protons comes inside this space, the space which is present between the both of two, that is the perimitochondrial space. So these protons travels inside it. Okay, these protons travels inside the perimitochondrial space, the space between the inner and the outer membrane. Now what happens, these protons move into the base of the oxysome. These protons move into the base of the oxysome. Okay, now these protons are moving inside it. You can see it over here. And now what is present over here, that is ATPase enzyme, which is responsible for ATP synthesis. Now what is the role of these protons, students? These protons helps ADP to combine with the inorganic phosphate and become ATP. Okay, these protons help ADP, adenosine diphosphate, to combine with an organic phosphate and becomes ATP adenosine triphosphate, and the enzyme which is responsible for the formation of ATP will be what? ATPase enzyme. Did you understand this thing, students? First, I am going to clear you this thing that ADP has two phosphate groups. Diphosphate means two phosphate groups. Now it requires only one phosphate to become tri. Tri means three phosphate group. So if it requires an inorganic phosphate, it requires protons also. So when the protons will enter this oxysome, when protons will enter, the inorganic phosphate will get activated, and this inorganic phosphate will get added to ADP, that is adenosine diphosphate. When adenosine diphosphate consumes or combines with inorganic phosphate, it will become adenosine triphosphate, and this is how our ATP is being synthesized inside the mitochondria. So the main function of the mitochondria is ATP synthesis. That is why it is known as powerhouse of the cell. Now did you got it? Okay, now I am moving towards the functions of mitochondria, which is going to be performed by the mitochondria in our body. So what are the functions? We are going to see it over here. The structure of oxysome, it is clear to you all. What happens? We will revise it shortly. The outer side, the inner mitochondrial membrane, oxysome is present. This proton here is accepted over here. This ADP combines with inorganic phosphate and becomes what? ATP. This is clearly understood to you. Okay. Now I am moving towards the next slide. Important facts about mitochondria. So first important fact about mitochondria, it is a semi-autonomous cell organelle. Semi-autonomous means it can replicate itself because it has its own DNA, its own proteins, and its own ribosomes. Okay, it has its own DNA, own proteins, and own ribosomes. And if a particular cell organelles have three of them, then it can replicate by itself. Now next point, only one mitochondria is present in the lower animals. Okay, lower animals or lower plants, only one mitochondria is present. So it is present, only one is present in the chlamydomonas. So chlamydomonas, you know that very well, it is a blue-green algae which I told you in the chloroplast part also, and chlorella. Okay, so remember this thing, only one mitochondria is present in chlamydomonas. Now the next point, 5 lakh mitochondria. 5 lakh mitochondria are present inside the flight muscle cell. Okay, did I told you about the flight muscle cell in the first part? Kolliker observed that it is present inside the insect cell. So 5 lakh mitochondria were seen inside the flight muscle cell. The next point, which is very important for us, that generally ribosomes are present inside the mitochondria are 70s. What the word I said? Generally. Generally means it does not mean that in every cell organelle the mitochondria will be like of 70s type, means the ribosomes will be like. It does not mean that the ribosomes will be 70s type in every mitochondria. I said the word generally here. Okay. So remember students, generally ribosomes are of 70s type. Here S stands for the sedimentation rate that we will discuss when we will study about ribosomal part. Okay. Now in mammalia cell, the ribosomes are present are of 55s type. Remember this thing student, in mammalia cell the ribosomes are of 55s type, or you can say the ribosomes are of 55s type. Remember this thing. In general they are of 70s type, but if we talk about mammalia cell, they are of 55s type. Okay, this is the great important thing you have to remember, and in many exams it is usually asked for general confusion because students over there do not think and they just take upon 70s, but you have to see that if it is written mammalia, then you have to take over 55s type. Okay. And I told you about that the matrix which is present inside the mitochondria is pale yellow in color, and that is due to riboferinine. Okay, the pigment which is present inside the matrix is known as riboflavin, and that is why it is also seen as pale yellow in color. Now, the mitochondria part has been over till now, means we have seen the important facts over here. Now, we will see the functions of mitochondria. So, first function of mitochondria is very well known. It is used for what? Energy production, means it is the storage battery of the cell, or we can say it is the powerhouse of the cell. So, its main function is to store the energy. Okay, so remember this thing. It stores the energy in the form of ATP, adenosine triphosphate. Next, it is responsible for cytoplasmic inheritance. Now, what do you mean by cytoplasmic inheritance, student? I am going to make you understand this thing over here. What is cytoplasmic inheritance, students? Cytoplasmic inheritance means the transfer of mitochondria from the hereditary material. Now, how it happens, students? You have learned about a little bit in fertilization chapters in past earlier classes. So, you will understand this thing, that if there is ovum present over here, so this is ovum, that is the female part, and this is the sperm, that is the male part, or male gamete we can say. Okay, the sperm and ovum, these are the male and female gametes. Now, what happens if there is a ovum and there is mitochondria present over here? This is the mitochondria which is present inside the ovum. Okay, now what happens? The sperm and ovum will get fused. Okay, and after getting fused, they will form what? A zygote. Okay, now what happens? The zygote contains the same mitochondria which was present in the ovum cell. Okay, the zygote contains the same mitochondria which was present inside the ovum. So, this mitochondria has been transferred from the female gamete towards the zygote. So, this is known as cytoplasmic inheritance. It means the male and the female gametes are responsible for transfer of mitochondria from themselves towards the zygote formation. Did you understand this thing? Now, we are moving on towards the next point. The next function of mitochondria. It is responsible for vitellogenesis. Vitellogenesis means yolk synthesis. Okay, yolk synthesis, you have understood it clearly, that mitochondria is responsible because there is a lot of ATP generation inside it. Okay, next point. It is a site of Krebs cycle for respiration. I told you I will explain it you in the upcoming further lectures when we will study about the human physiology, the cellular respiration. Now just you have to remember this thing, that it is responsible for the cellular respiration or the Krebs cycle in the living organisms, because the oxysome contains the ATP production enzyme ATPase, which I told you in the oxysomal diagram. Next point. Now, we are over with the mitochondria, the functions of mitochondria, the structure of mitochondria. Now, we are going to discuss about the single membranous cell organelle, that is endoplasmic reticulum. An endoplasmic reticulum is abbreviated in short form as ER, and then it is divided into two further types. That is, first is the smooth endoplasmic reticulum and second one is the rough endoplasmic reticulum. Okay. So, what are the functions of reticulum and where are they found inside the cell and where they are present and how they are formed, we are going to discuss in this upcoming slides. Okay. So, first you have to remember this thing, student, that what is endoplasmic reticulum. It is also known as ER in short. Divided into two categories. The first is smooth endoplasmic reticulum and the second one is rough endoplasmic reticulum. Now the students are little bit confused that why ma'am is saying rough and smooth. It is based on the texture. No students, it is not based on the texture. It is based upon the ribosome present. Okay, if the ribosomes are present on the surface of the endoplasmic reticulum, then it is considered as rough endoplasmic reticulum. And if they are absent, then they are known as smooth endoplasmic reticulum. Means if we see the structure here, then if it is like this, and if there are ribosomes embedded on the surface of the endoplasmic reticulum, so it is known as what? RER. And if it is absent, means it is of smooth shape, then it is known as SER. Now moving on towards the next slide, we are going to discuss about the endoplasmic reticulum, that it is a network of membranes which is present in the cytoplasm. Now you can see it clearly here, students. You can imagine in your mind that when the nucleus is present, the nuclear membrane has protrusions, and these protrusions form the endoplasmic reticulum. This endoplasmic reticulum, which is formed by the nuclear protrusion, is known as what? Rough endoplasmic reticulum. And after rough endoplasmic reticulum, the smooth endoplasmic reticulum is present. Okay. Next point about this, it was discovered by the three scientists. First is Potter, second one is Claude, and third one is Fullan. Okay. Next point, it is present in all cells except prokaryotes. As I told you, in mitochondria also they were primitive, so they were not considered in the upcoming cell organelles. Okay, so everything which is main important and used for protein synthesis or ATP synthesis, so they are absent in prokaryotic cells. Okay, and it is also absent in the mammalian RBCs. As I told you, the mature RBCs lose all the cell organelles which is present inside it. So mitochondria as well as endoplasmic reticulum, both are absent inside the RBCs, that is erythrocytes of the mammalian. So mammalian erythrocyte, it is absent. You have to remember this thing. Put a star mark over this line. Now the next, we are going to see the three components of ER. Now the ER, endoplasmic reticulum, is divided into three components over here. So first one, the first part will be cisternae, second part will be vesicles, and the third part will be tubules. Okay, so first part will be cisternae, second part will be vesicles, and the third part will be tubules. Now you can see the diagrams over here which I have put on the slide. You can see the lamellar structure which is present over here. This is the cisternae. These are the small lamellar tubes which are interconnected together like this. Okay. These are interconnected together like this. So these are the lamellar structures, and they have the embedding of ribosomes over them. So these are the ribosomes which are present over it. Okay, these are known as cisternae. Next one is the vesicle part. Vesicle part is like rounded shape, small rounded shape, and they also have ribosomes embedded over them. You can see here, students, clearly. These are the vesicles. Okay, these are the ribosomes present over them. Now if we talk about tubules, tubules do not contain ribosomes. Okay, they do not have the embedding of ribosomes. They are smooth. Okay, so the tubules are like this. Okay, so you can see the surface over here, it is smooth, means they do not have ribosomes. So smooth and rough means this only. If ribosomes are embedded, then they are categorized as rough, and if they are not, they are present, then it is categorized as smooth. Okay. Now the three components we are going to discuss it over here, the cisternae, vesicles, and tubules. Okay, now if we talk about cisternae, they are long, flattened, parallel arranged structures. You can see the diagrams over here. Okay, these are the long, flattened, arranged structures over here. This type of arrangement you can see. Okay, they have embedding of ribosomes. They have embedding of ribosomes, and they are active in protein synthesis. They are active in protein synthesis. Means they have 40 to 50 diameter. Okay. Now if we talk about vesicles, they are small spherical in shape. Okay, they are small spherical in shape. They have ribosomes over them, and they are found in synthetically active cells. Means they are found in those cells which are synthetically active. Now the last part, which is the tubular part. Okay, tubular part has smooth walled. As you see, the ribosomes are absent, so it is what? Smooth walled. Okay, and tubular spaces are present over there. They are found in non-secretory as well as steroid synthesizing cells. Okay, they are found in non-secretory as well as steroid synthesizing cells. Steroid synthesizing cells means the cells which produces the female and the male hormones like testosterone and estrogens. Okay, now did you understand the three parts of the ER? Now we are moving on towards the next slide, which is the diagram of the ER. Now we are going to discuss it over here. There is a nucleus, and if you see, there is a nuclear membrane, and nuclear membrane when invaginates means the protrusions which are formed by the nuclear membrane are considered as rough endoplasmic reticulum, and they have ribosomal heads present over there. Okay, now it is providing a great space. You can see over here it is providing a surface area to the cell because it has large surface area. Now this part which do not contain any kind of ribosome is considered as smooth endoplasmic reticulum. So you can see the differentiation over here in this diagram. If the ribosomes are present on the part of the nuclear membrane which is being protruded inside the cytoplasm, then you can see over here that it is considered as rough endoplasmic reticulum. Now if there is no ribosome present, then they are known as smooth endoplasmic reticulum. And next, the labeling of other things are also there, that there is secretory vesicle present which secretes the hormones, as I told you. The lysosome is present, which we are going to discuss on the upcoming slide, lysosome, Golgi apparatus. Okay, and if we talk about nucleus, we have discussed it very carefully that nucleus is what? It is the main head of the cell, and the nuclear membrane when produced, it's formed the endoplasmic reticulum. If ribosomes are embedded over it, then it is considered as rough, and if it is not present or absent, then it is considered as smooth. Now did you understand the structure of endoplasmic reticulum? Moving on towards our next slide. We are going to discuss the differentiation between the smooth endoplasmic reticulum and the rough endoplasmic reticulum. So what are the great differences between both of the two? First, you have to see that the smooth endoplasmic reticulum is made up of only tubules. You have seen that it is made up of only tubules. It has only a tubular structure like this. Okay, now these are made up of cisternae and vesicles. Why they are made up of? Because you have seen that in cisternae and vesicle, in both of the two parts, the ribosomes were present. Now moving on towards the next point, ribosomes will obviously be absent inside the SER, over the SER, and for RER it is present. Position, it is near the cell membrane, and you have seen that it is near the nucleus. Why it is near the nucleus? Because the nuclear membrane itself protrudes and forms the endoplasmic reticulum, the rough endoplasmic reticulum. Now what are the functions of rough endoplasmic or smooth? First we are going to talk about smooth. It is responsible for the synthesis of lipids, proteins and steroids. Okay, steroids means the male and the female hormones, that is estrogen, progesterone, testosterone, and it is also helping in membrane biogenesis. Now what is membrane biogenesis, student? The formation of lipids and proteins for the membrane, cell membrane formation is known as membrane biogenesis. Now if we talk about rough endoplasmic reticulum, the main function of rough endoplasmic reticulum, it helps in the protein synthesis. So this is the main important function. Now if we talk about the occurrence, where it is found, so it is found in the fat cells, that is the adipocytes, means the fat cells or the cells which contains fat in the every living cell organelle. Okay, so the fat cells, which are present inside every living cell organism, there are present the SER. And if we talk about RER, they are found in mainly the protein forming cells like Nissls granules of the nerve cell. Now, what are the Nissls granules of the nerve cell? See students, here I am going to tell you about the Nissls granules. So, this is the diagram of a neuron. Okay, this is the diagram of neuron. I am going to draw it clearly so you can understand it better well. Okay, and these are the axons which are present over it. You can know it very well, and in lower classes also you have studied about this thing where it is present. This is the nerve cell which is present inside the brain, and you know this thing. This is the largest cell also. I have told you in the first lecture. Please do not forget anything about the first lecture. Now these are the small dark stained parts which are present inside the neuron. Okay, so these are known as what? Nissls granules. Didn't you understand this thing? Okay. Now you can see here it is used for the formation of protein inside the Nissls granules of the nerve cell. Now we are over with the difference between the two. Okay. Now next slide. Now moving towards the next slide, what are the functions of endoplasmic reticulum? As I told you, it acquires a large surface area, so it increases the surface area of the cell as well. So first point is about that it increases the surface area of the cytoplasm for various metabolic activities. Now the second point, is about that it provides the internal support. As it is a large surface area, so it also balances the cell, so it provides the internal support to the colloidal matrix of the cell or the cytoplasm of the cell. Next point, it acts as an intracellular transport system. Means you know that the ribosomes are embedded over the endoplasmic reticulum and responsible for protein synthesis also. So in Golgi body we will discuss it more about this thing, that how the proteins has been synthesized and what is meant by transcription, translation, and the things which are formed inside it are transported from one cell organelle to another cell organelle, and ER is responsible for that thing. Okay, for various substances like proteins and lipids. Material synthesized can be stored in different parts of ER. As you say that the lamellar structures or the tubules or the vesicles are present in the internal part of the ER, so the materials which are being synthesized can be stored in those parts only. The next point is the protein synthesis that occurs in the ribosomal part which is embedded on the ER surface. You have seen that this is the part over here, and this is the ribosome embedded. Now the proteins has been synthesized over this part, and these proteins are either used by the cell or exported out in the cytoplasm for the use for other cells. Okay, now next point and the last point about function of endoplasmic reticulum, which is very important, is that it releases the secretory hormones, that is testosterone and estrogen, which are the female and the male hormones. Okay, so it helps in the synthesis of sex hormones. Okay, it helps in the synthesis of sex hormones, that is testosterone or estrogen, which is present inside the female and the male body. Okay, so progesterone in female body, or progesterone or estrogen in female body, and testosterone in male body. Okay, so this one is the last part of the endoplasmic reticulum. Now I am going to do a quick revision of endoplasmic reticulum. Where the endoplasmic reticulum is present, the nuclear membrane protrudes and form the endoplasmic reticulum. The first will be rough because the ribosomes are embedded over it, and the part after that will be smooth endoplasmic reticulum because the ribosomes are absent. Now the functions we have read about both of the two. Now the main function of endoplasmic reticulum is protein synthesis and the synthesis of the sex hormones. Okay. Now I giving a small assignment to you all to do. Okay, this is just a small assignment from my side, so please do it. Okay, and these questions will be discussed in the upcoming next lectures, and I don't think so any problem will arise. Please solve the module carefully. Okay, thank you.